Hello, everybody, and welcome to this webinar. We're just going to give a couple of moments here to allow a couple more attendees to trickle in, and then we'll get to the good stuff here. So please be patient for a couple more seconds here. All right, I see a couple more people have logged on here. It's past 10, and I like to be on time, so let's get started here. So my name is Will Olds. I'm the scientific officer here at Protein Tech. I've been in this position for about 5 years, and it's been an exciting ride so far. And I'm delighted to talk to you guys about some of our newly developed cytokines, antibodies, and ELISAs to investigate the novel coronavirus. I do want to mention that we do have some handouts available. It should be on your control panel, and just talking a little bit about Protein Tech, as well as like kind of our overall COVID-19 flyer that has information on what you could use. And so let's get started here. All right, so who is Protein Tech? Who are these people? Who is this person that's even talking to you? So Protein Tech has been around since 2002. We have an antibody target range of about 13,000. That represents both rabbit polyclonal and mouse monoclonal antibodies, as well as some newer recombinant antibodies. We have 200 plus ELISA kits. We have human cell-expressed cytokines and growth factors. We have directly conjugated antibodies for your staining experiments, as well as neutralizing antibodies and some pre-stained protein markers as well for your Western blots. So something that's great about Protein Tech is that everything is 100% manufactured and validated in-house. We have all the ISO certifications for your clinical trial and therapeutic needs. We have 5 sites globally with full product inventory, so you never have to wait. And we have products, and our products have been cited over 65,000 times in every publication, every journal we can think about, as well as having some GMP grade products available. And we love to really support the scientific community, like giving webinars like this, as well as giving mentorship awards, travel grants, and other technical workshops. So this figure right here really kind of represents who we are as a company. So our slogan is really from our bench to your bench. And so unlike a lot of other antibody vendors, we are not resellers. Everything we make, we send directly to you, as you can see from this line, from us to you. And what that means for you as a customer is that we're able to really have a lot of quality control over our products, and we're able to really give an idea of kind of a stable supply of then, you know, the people who make the antibody and people giving you the antibody. And so I like to say that validation is our middle name. So we have a stringent validation process. So before everybody was even talking about the pillars of validation, we were actually launching our siRNA knockdown validation initiative in 2015. And in 2016, we won CyDAB's award for most exciting antibody validation initiative for this effort. And we have the broadest coverage of antibody targets with specificity verified by KDKL validation. So it's over 2,500 targets at this point. And as well as having antibody-specific protocols available. So you get to see our transcribed lab notebook pages. And so we want to it's not about us, it's about you. And so we're really excited to help people to publish in over 65,000 publications, and really to be a growing effort to really accelerate your research and help you find the cures and other exciting biology. All right, so let's think back to our history here. So in terms of diseases that we studied, we've been looking at cancer since at least 1600 BCE. All right, so that's about 3,600 years that we have been looking at cancer and been studying it, trying to figure it out. Since about 400 BCE, we've been trying to conquer influenza. And in 300 BCE, we've formerly been studying biology. And then at about 100, and then since about 150 CE, we've been studying anatomy, how the different organs are stitched together and help us grow and proliferate. So, and we've only been studying SARS and MERS since about 2000s. And the coronavirus is, as you can see, all the way in right here, about 5 months ago. And so think about how much we know about cancer, the flu, biology, anatomy, even SARS. And we have a lot of catching up to do to really get up to the sophistication that we know about all these other diseases. Since January, according to PubMed, more than 11,000 papers have been published on the novel coronavirus. However, critical questions still remain. Why are some patients asymptomatic? How long does acquired immunity last? Is antibody-dependent enhancement a possible mechanism during infection? How many organs are infected? And then how many organs are infected? And what is the cause of the reported kidney, heart, and neurological long-term issues? So our goal today is to empower you with antibodies, ELISAs, and proteins to understand COVID-19. So this figure here kind of represents the relatively little that we know about the coronavirus. So as you're probably well aware, everybody's favorite receptors these days, ACE2, binds to the coronavirus, as well as with some help of TMPRSS2 to help this enter. This then fuses into an endosome, and it's uncoded. There's a lot of transcription translation goes along, goes to the ER, and eventually gets assembled into the new viral particle and goes out. All of the stuff that happens here is relatively unknown, and that's what we really want to help you find out with our antibodies. So there are a couple of targets I want to highlight here. So obviously ACE2 and TMPRSS2, as well as some really interesting ones that are in kind of this unknown space in the middle here. All right, so ACE2, as I said, everybody's favorite antibody at this moment. So we offer that in a rabbit polyclonal and a mouse monoclonal form. The rabbit polyclonal has about 5 publications as of now. It's been validated by KD/KO validation, as well as being useful in a variety of applications, including Western blot, IP, IHC, immunofluorescence, and flow cytometry. We also have a mouse monoclonal antibody, which is depicted here on the right in Western blot. Using PNJSF, you can see how the band slides down quite a bit after adding PNJSF, which helps show that this is specific for the ACE2 target. All right, so now the next target I want to highlight is TMPRSS2. This is a rapid polyclonal antibody, and it's been validated in both Western blot and IHC. Some examples are here on the right. Western blot is from colorectal 220 cells, and then this IHC is from human prostate cancer. All right, so one of the kind of more interesting targets I want to highlight is cathepsin L. This is this antibody that we offer is a rabbit polyclonal, been KD/KO validated. And what's kind of interesting is that this has already been shown, this target has already been shown to play a role in SARS and MERS pathogenesis. So, you know, it may also play a role in SARS-CoV-2 as well. And on the right here, we have an siRNA experiment showing that when you add an siRNA construct against cathepsin, the band disappears, showing that this is specific. The next interesting target I want to highlight is MX1. This has got 52 publications. It's got KD/KO validated, and this one interferes with general viral protein translation. Could this, this target could be implicated within the SARS-CoV-2, and it would be an interesting one to check out to see if it has an effect. Another target I want to highlight here is ISG15. This antibody has been cited 18 times in KDKO validated. And again, this has general antiviral activity. Again, this may be something that we could take advantage of in terms of therapeutics or in terms of the pathology of this. All right, so now you've got a bunch of antibodies and you start to run into a problem pretty quickly, right? So you have a stain for your nuclear, stain with DAPI, you have F-actin, you can stain with that, and then you try to stain for a bunch of other targets and you run into some issues. So you have a mouse monoclonal, and that's great. You're able to get a nice secondary to also latch on to this antibody. You get some nice signal. And then you have a rabbit polyclonal towards this protein. You get a nice secondary for that. However, for this other one, you're only able to find a rabbit polyclonal. And so you can't just use their secondary on there, right? Because then the secondary is going to get this guy as well. And so that becomes an issue when you want to do more than just a couple of targets and a couple of targets and use more than a couple of antibodies on a specific tissue. And so to help scientists with this, we have created a line of conjugated antibodies, and so we call these Coralites. This is a proprietary dye that we have created, and it comes in a couple of different flavors with wavelengths you've heard of before. So the first one's Coralite 488, Coralite 594, and lastly, Coralite 647. And so they're really great for these immunofluorescence studies requiring multiplex co-labeling studies, and you don't need to use any secondaries. So this background here is great HepG2 cell immunostaining using ATP5A1, cytokeratin, and DAPI, making this beautiful staining over here. And so what are the Coralite antibodies and what is this dye? So these are all of our antibodies that have been validated and published before, now conveniently conjugated to multiple colors and are ready to use. So we already know the base antibody is very good, and so we just add a fluorescent dye to it, and now it's perfect and validated and ready for use. So Coralite itself is a proprietary fluorescent dye, and it is equivalent to ELISA dyes in performance, in brightness, stability, and size. And so currently we have about 400 targets that have been conjugated with a variety of these Coralite dyes, and they're growing. So in terms of the targets that we've selected thus far, we've picked ones mostly so far in immunology, neuroscience, and cancer. And on the right here, you can see some an example of one of our neuroscience targets with a TUB3 antibody, which is a neural specific marker, and you can see this beautiful neuron staining here. And so we have validated these for IHF as well as ICC as well. And as I mentioned before, these are all previously cited in major journals, and most have KDKO validation already. And we're adding to this portfolio every month. So as I mentioned, we started with some published KDKO antibodies, such as this one over here, the CD206 antibody. We add the Coralite dye to it. And now we get the magical, beautiful looking stain that we see here with the in green with CD206. So I just want to show some great pictures and usages of the Coralite product so far. So on the left here is an immunofluorescent picture here of HepG2 cells with the ATP5A1 and cytokeratin 18, which you previously saw as the background, but now it's in the foreground here. And on the left here is a great staining of rat brain with GFAP, a glial marker, and MAP2. As well as DAPI in there as well, and creates this beautiful staining that you see here. So when should you use Coralite antibodies? When will they perform the best? So they're really great for multiplex imaging, as I said before. They're also really great for using for cell markers, organelle labeling, or for highly concentrated proteins. Here are some examples of some markers here. This is a cytokeratin 18 for the cytoskeleton. And on the right is GFAP glial markers, as I said before. Great Coralites great for that. These are abundant markers, and they can make some very great images. They're great for organelles as well. And so on the left here is staining with DAPI and lamin B1. Lamin B1 sells the nuclear membrane, so you can see that kind of hovering around the DAPI staining here. And on the right here, we have HepG2 cells stained with COX-4, which is a mitochondrial marker. And so the Coralite antibodies are really great for concentrated proteins as well. So here in A, B, and C, we have staining with two different markers for the cilia here. So one's RL13B, which is in red, and then there's acetyltubulin 48. And these both mark the same thing. And as you can see here, in the green and the red, they overlap completely and make this a really great staining here. And so these Coralite antibodies are, this direct staining is really great, but it's not perfect. Indirect IHF is best for low expressed proteins. So these Coralite antibodies, can bind to these targets and fluoresce quite significantly. However, the indirect, you're getting the secondary on there. It's amplifying the signal significantly, and so you are able to get much brighter signal and able to amplify the signal of a low abundance protein. Okay, so back to our little schematic here. So now we've got a bunch of antibodies looking at different targets. We've got some Coralites in there as well. But the guy that we really want to track throughout its life cycle here is the SARS-CoV-2 itself. And so we've recently entered into a collaboration with Active Motif to provide recombinant coronavirus antibodies towards the spike protein. And so these are antibodies that have been derived from B cells from recovered COVID-19 patients overseas, and they've been screened for binding affinity towards the S protein. And after they've been screened, they were cloned and expressed in Hec9 293 cells with a human backbone. And because these are absolutely brand new, as of maybe a couple of months ago, there's been limited characterization so far of these antibodies. We offer 9 clones as of this moment, and they have been tested in several different applications, the Western blot, neutralization of the coronavirus. And we offer all 9 of these clones for you to kind of test and see and kind of test the limits of what these antibodies can do. Recently, a few clones have been shown to have neutralization activity. And so this is a paper that was published, that was in the bioRxiv a couple of weeks ago, showing some neutralization of a pseudovirus infection model. So some of the advantages of this line is that they have a human backbone. So they can be easily multiplexed with rabbit and mouse antibodies. Some of them have neutralizing abilities for in vitro and potential in vivo studies, as well as being useful for assay and testing development. We obviously need a lot more of these serology tests at the moment. So speaking of serology tests, we do offer IgAs and IgMs for use, and IgGs for creating these serology tests for expanding the contact tracing and seeing who has immunity towards this virus at the moment. Okay, so now we've got a bunch of antibodies to play with here to really find out who's all playing with the novel coronavirus. And there has been some research showing that CAR T cells may be useful in the fight against the coronavirus. So as you guys are well aware, the CAR T cell therapy starts with collecting patients' white blood cells, doing some CRISPR trickery in there, expanding the T cells, and then putting them back into the patient. So this kind of part in the middle here, where you're engineering the CAR T cells and you're growing them, and you're maintaining them, you need the cells need a lot of cytokines and growth factors in order to live and proliferate and be maintained. And so we offer a line of human HEC293 cell expressed cytokines and growth factors for this purpose. And so what are the advantages of that? So most protein production is done in E. coli. So following the central dogma here of DNA, goes into RNA, and then is made into protein. For E. coli, it just hops right off the ribosome. You add the purification tag, and then there's a bunch of endotoxin there as well because it's E. coli. That's a bit of a problem, especially if you wanted to make a human protein. So for humans, these proteins generally go to the ER, where the ER chaperones help shape these proteins. There's been several maturation processes that occur, and eventually get glycosylation, dimerization, and what have you. So this is very complex. This is a complexity that our HEC293 cell express system has that E. coli just do not have. And we have an ISO 1345 certified GMP facility. These products are incredibly safe because they are animal component and xenobiotic free. So you don't have to worry about any of these endotoxins, any of these other issues that happen with using E. coli in creating proteins. And because they go through all the post-translational modifications and maturation, they have native folding, as well as having native glycosylation, resulting in high stability and activity. All right, so one of the products I want to highlight here is our GMP grade Activin A. And so this is one that is essential for stem cell maintenance and for cell culture maintenance in general. And this is a protein that's highly processed. So if you just look at this gel over here, so the Activin A precursor is pretty heavy. It's processed later on into a monomer, and then finally into a dimer. And so as you can see here, comparing this CHO cell expressed one versus the humankind Activin A, you can see that there's a thicker, more significant band for the dimers here, showing that this is the mature active form. And currently, this is the only GMP human cell expressed Activin A on the market. We also offer FGF basic thermostable. And so here is an experiment showing the stability of FGF basic in xeno-free media without cells, and showing that over time, the FGF basic that comes from that we make in red here lasts for several days, whereas the one that's made from an E. coli expression system, it is practically zero after 1 day. And so what this means is that you get a little bit more bang for your buck, and as well as not having to go in on the weekends to change your cells. All right, so now something else that's been a bit of a concern for the COVID-19 is the cytokine storm that happens in a lot of deadly cases. So we also have some antibodies towards those as well that we call Neutrokine antibodies. And so these are used, and so we make these using our humankind proteins by injecting them into the mice, and then selecting for antibodies that come out of this mouse that are able to disrupt the binding of the cytokine to the receptor and causing the cytokine storm. So we have about 9 of these right now, and they're growing every couple of months here. And so one of the ones I want to highlight here is our TNF alpha antibody. And so this is a neutralization assay here depicted. It's a bit confusing, so let me just walk you through how it works here. So let's start here with this green line. So this green line is just adding the the protein itself to the plate and seeing what happens to the cells that are under study. And so in this case, it's TF1 human cells. And so as you can see, the proliferation it goes up significantly as you add more protein, which is as to be expected. And now I want you to take a look at the activity here when you start increasing the amount of antibodies. So here's a leading competitor. And so as you add more of this antibody, it decreases the amount of proliferation. However, you notice that this curve, the one using a protein text Nutrikine antibody, the curve is much steeper and it goes down to very low activity, very low proliferation very quickly. So this antibody has a much higher affinity and much higher neutralization activity than this competitor here. Okay, so now let's look more into quantifying the products that are within the cytokine storm. So we've created a line of ELISA kits that we call Cytokine ELISA kits. And again, like before, like the Nutrikines, these are made using our humankind products. So the humankind antibody or humankind proteins are injected into the mouse, and then we select 2 antibodies from the mouse in order to make the ELISA, in order to make a sandwich ELISA kit. And so below here, I have a little table here just showing our sensitivity in our ELISA kits compared to their competitors. And for INF gamma and cystatin C, we have 20x and 10x fold better sensitivity than competitors. One of the ones I want to highlight is IL-6. Obviously, this is one of the major players in the immune response, and so this is made using our IL-6 humankind protein cytokine. So this is a sandwich ELISA. It's got a great sensitivity of 1.2 picograms per mil, as well as having a significant range of 7.8 to 500 picograms per mil. All right, so now you have antibodies to detect the coronavirus, to look at what's happening with ACE2 and TMPRSS2, as well as investigating other targets, being able to multiplex with the Coralite antibodies, being able to help expand your CAR T cells for CAR T cell therapy, as well as trying to neutralize or trying to understand more about the cytokine storm as well here. And so I want to thank you for attending this webinar, and I am open for any questions. Feel free to email me or ask questions now. So I'll just keep it open for a couple of moments here. I see one person that's asked about the audio is cut out for them. The recording is going to be shared in case there was some issues with the audio here. All right, I don't see any other questions, but feel free to email me at will@ppglab.com if you have any more questions. Thank you guys for attending, and happy hunting. Take care.